So, good evening, everyone. Welcome to the 78th session of the Beyond Research series, where curiosity meets the conversation, and a series by Biopractify. And today's session is titled as Journey of a Researcher. So, it's my pleasure to introduce our distinguished speaker, Dr. Sugandha Asthana. She's a biotechnology researcher, entrepreneur based in Pennsylvania, USA. She holds a PhD in biotechnology from MIT University, where her research focused on engineering bacterial metabolic pathway for sustainable biofuel production. She also earned an MSc in molecular and human genetics from Banaras Hindu University and a bachelor's degree in microbiology from University of Delhi. Driven by her passion for making scientific knowledge more accessible, she founded Simplify Research, an AI-powered platform that transformed complex research papers into a structured, summarized, plain language explanation methodology, visualization, flowchart, and research roadmap. Her work is helping students to and researchers, professionals understand the and engage with scientific literature more efficiently. Today, we will share her expertise at the intersection of biotechnology and artificial intelligence. Please join me in extending a warm welcome to Dr. Sugandha Asthana. Thank you.
Hi everyone. Let me just start with sharing my screen and we can begin. Give me 1 second. Hi, is my screen visible? Okay, so let me just welcome you all to today's talk for Biopractify, which is basically about how a research journey goes from your bachelor's all the way to your PhD. So the topic of today's conversation is going to be journey of a researcher, and I want this to be an interactive session. It just, you can ask me questions. Even in the middle of the presentation, we can have a conversation about something that you don't understand, or if you want to, clarify something between the PPT, please feel free to interrupt me and ask me anything that you feel like. So thank you for the great introduction. So as mentioned, I am Dr. Sugandha Asthana, and we are going to cover 6 basic topics today in this talk. So first, it is going to be what kind of academic path that I took and where it has taken me till now. How do we work out the PhD admissions, especially in India? What kind of different exams there are? How do you prepare for it? What kind of things that you need to take in mind before entering the exam hall or even the interview hall? Then a little bit of the detour I did from my scientific journey, and I worked with an industry for a couple of months to figure out whether it's the right path for me or not. And what happens during your PhD research, how things work out, what kind of problem questions that you need to answer, and different kind of options that you can explore after your PhD. Doesn't just have to be academia. There are a lot of things that you can go through. And finally, the product that I am currently working on, which is Simplify Research. So taking some inspiration from the famous movie Durandar, we are going to start chapter wise, which is chapter 1, which is the academic path. So this was my academic journey. As you can see, it started off in 2010 and it is still kind of ongoing. So I did my bachelor's in Delhi University in BSc Microbiology. Then I moved on to my master's, which was from Banaras Hindu University in Molecular and Human Genetics. Then I did 2 years of PhD at IIT Kanpur from 2015 to 2017. But, somehow life gets in the way and things don't end up working out. So, I had to drop out of that. And for a brief period of time, I did the industry work, which I mentioned, which was a pathology lab, and I was in charge of marketing there. And then finally, I went ahead and rejoined my PhD because, I thought that research is the way to go. And finally, after finishing my PhD, I went into trying to get into entrepreneurship. It is just beginning. So I'm trying to still figure out how things work and where do I land and how to process things further. So um, this is just a brief explanation of what kind of different MSc exams do we have in India. So this is basically for the science stream. I personally gave three exams rather, TIFR, JGBLs, and CUET-PG. Most of the universities, they are open to all the entrance exams that I have mentioned. And if you end up clearing that, then there is a counseling session that happens in the university, which you can figure out what kind of subjects you want to take, what kind of expertise that particular university have, and then you can choose your master subject. So I was able to clear the CUET PG for Banaras Hindu University, which is why I chose the discipline. Although my bachelor's was in microbiology and they do have a microbiology department, but my interest was more towards genetics. So I did decide to choose molecular and human genetics in Banaras Hindu University. So this was my work for my tenure at BHU. We worked basically for my dissertation, which is the last 6 months of your master's. We worked with neutrophils. So the guide that I was working under, her focus was on maternal health. So she realize that there are certain conditions in which when the baby is born, they're basically low birth weight with no fault of the mother, like she might not be getting enough nutrients because of her financial background or any other health conditions. So how do we help the newborn baby that is being born with such conditions? So she noticed that because the low weight birth babies are born, their immune system is also not functioning properly. Either it is too advanced or it is not working at all. So she focused on neutrophils. So she asked me to work on this along with a couple other PhD students that were there at that time. And she is working on a new method. I'm not sure if you have ever heard of this, that neutrophils undergo immune response, which is called netosis. So netosis is basically a burst of neutrophil. They don't survive this mechanism, and they end up secreting all the interleukins and chemokines into the environment so as to stop the infection from spreading within the cell or within the environment. As you can see in the image, if you can see this thin line between connecting two cells, that's how nets look like between neutrophils. This is basically an image of a neutrophil seen under the microscope, and it is stained with DAPI. So this kind of net formation releases the chemokines into the environment, helps fight off the infection, but it has been noticed that in certain conditions this is overexpressed. So once overexpressed occurs, it has lead to problems like sepsis, lupus, even thrombosis. So we were working on how to curb the overexpression of netosis, and to do this, we used we first work with gene knockout attempt. Okay, so being an MSc student, this was my first exposure to lab experiments, like proper wet lab experiments, and a lot of thought and a lot of planning goes into it before you actually start your experiments. So just to finish a dissertation of just 6 months, you have to plan out the chemicals that you need, the kind of work that you need to do, and the proposed hypothesis that is in your brain. It should be short enough that you are able to finish it within the 6 months, and it should be important enough that, if you have worked on it, people should appreciate your work. So, of course, because you are a dissertation student, you are always working with a PhD student, and they have most of the things figured out because, of course, this is part of their journey as well. So you get to learn a lot during your dissertation. It is not just about the science that you are working, it is also about understanding that there are multiple things that can go wrong, and how do you actually work on it so that it gets to a stage where you are able to present it after 6 months. And of course, 6 months is a very short amount of time, not a lot of work. Actual work can get into it if you're doing a wet lab experiment. There are multiple things that goes wrong. There are setbacks. Sometimes the chemical is late. Sometimes the machine breaks down, and you are some things are just beyond your control. So when you are starting your journey as a researcher, my first and foremost advice would be is to be patient. Okay, so things may not work out and you might get frustrated, but they do have a way of working out at the end. So be patient, be calm, um, try to have as many options as you can when you're working on a problem. So if method A doesn't work out, then you can go for method B. And if method B doesn't work out, you can go for method C. So as you can see, when we were trying to figure out how do we suppress this netosis in certain conditions, we first attempted a gene knockout for LTB4R, which is the gene for interest. So this we tried this for roughly a month or two, and we didn't get any colonies. We tried different methods for cloning. We even tried electroporation method, but that also didn't work. So we figured out that if gene knockout is not working, let's go for knockdown approaches. So knockdown is basically you are not deleting the gene from the system, but you are essentially turning the gene off. So we did do that. We used siRNAs for that, and that ended up working in our favor. And we noticed that once we gave a 50 picomole of siRNA, there was an inhibition of the gene, and this suppressed the netosis formation. So this was just stage one of how things can work. This was what I worked on, and of course, the PhD student who was working with me ended up doing a lot more work on it, and he ended up figuring out how do we actually if we can translate this particular process to industry level. So I worked on a very small portion of it. So this was just like inhibition of one gene to study its expression and to see whether it is actually working in stopping netosis or not. So this was my PhD dissertation, and as you can see, we used HL60 cell lines. We worked with flow cytometry. I also worked with Western blot. There was immunofluorescence involved, as well as confocal microscopy. So this was just like a bunch of techniques that I had never ever used before, and I had to use. And even the PhD student who was working with me had not used a lot of techniques. So both of us sat down and figured out a lot of things during this time. So this was like my first introduction, and it was honestly very fun, though frustrating, and which led me to start my journey towards my PhD because I wanted to, get more into the research part of it. So, this is all about moving on to chapter two, which is PhD admissions. So we have four major exams in India. First is CSIRF. This is and second is GATE. Third is DBT, and fourth is again RET, which is your university specific examination. So for the first three, CSIR, GATE, and DBT, there are online examination, and they directly move you towards getting a fellowship for your PhD. Usually starts off with 35,000, 37,000 rupees a month, and then moves on when you end up becoming an SRF. So the pattern for both all three exams is completely different. For CSIR, it is mostly core knowledge questions. You will have one section with aptitude tests, and the rest of the two will be knowledge based. You have to pick your subject of whichever is your strong suit. So they have a lot of a long list of subjects that you can choose from. With GATE, you this is basically an exam which lets you get into all the IITs, ISCs, and even certain central institutes like ISR. For GATE, for if you're in sciences, there are two subjects that you can choose from. That is, one is life sciences, and the other one is biotech. So I chose life sciences because the kind of syllabus that they had matched my previous degree, which was my bachelor's and master's. So I ended up choosing life sciences. And for DBT, this is basically biotech. It is all short answer questions, long answer questions. Mostly, I have noticed that they don't have a lot of application based question, but more knowledge based question. Whereas CSIR and GATE will have a mix of both. CSIR in section C will have a long paragraph experimental question that you have to solve. So with CSIR, when you are trying to pass through this exam, you have to make sure that you are within the time limit, because sometimes the paper is of 3 hours, but then considering the questions are so long, you tend to overlook the time part and miss out the questions towards the end. With GATE, although you have a plenty of time, there will be questions that you will need to solve. Like they'll have maths question. If you have life sciences, you'll have a chemistry section that they'll ask you questions, per mole, how much energy is required, or the kind of questions like, if a reaction is taking place, what is kilojoules that will be needed to process the reaction. So you have to solve these questions. So once you're solving, things tend to take a lot of time. So for each of these exams, my suggestion is to sit down, time yourself, and then look up previous papers that are already there, and answer all the questions. Okay. Once you start doing this, you will realize there is a pattern to these questions. Each paper for CSIR, GATE, even DBT, they have a pattern. They'll have certain ways that they move through the paper. And after, if you go through all the past papers, at least 10 years, you will see the university that sets the paper each time, and that is not for CSIR, especially GATE. You will see certain types of question are prevalent when certain university is setting that paper. And this information is publicly available. It's not like hidden somewhere. So if you're sitting in the GATE examination or DBT examination this year, you need to go and look at who's setting the paper, and go back a couple of years and see what kind of questions did they set. And sometimes some questions overlap. So what happens when you end up doing this, you increase your chance of scoring better. So for example, even if out of 10 questions you are able to get two, so you are damn sure that these two questions are correct because you would have already solved it. So even if you get two questions correct, you move up your rank a lot. By 1,000, you can move up. So just from looking at the past papers, you will be able to figure out what kind of questions they will have. You will be better prepared. And since you would have timed yourself, you would know how fast you need to go, and if you want to skip a certain questions and leave it towards the end, then you can do that. With the institute examinations, it is changing every year. So every university sets up their own paper. They have their own entrance test, and that entrance test usually changes every time. And mostly I've noticed that these questions are also not available online. So another way to look at it is just check out what kind of work is being done in that particular university. So for example, if I'm from Amity and I can look at what kind of labs Amity has, so mostly the question paper that they'll design would be depending on the kind of work that is already being done in the university. So you can focus more on those kind of topics rather than just reading everything and everywhere. um, the topic is the syllabus is very vast for all these exams. You don't want to get lost in topics, subheadings, notes and everything. The better way to prepare yourself for these exams is to look at the papers. There is another way. You can also approach students who are working in the university, who are already in the PhD program, and more often than not, these students end up replying, and you can have a one-on-one conversation. You can ask them what kind of questions they had, the kind of interview process the particular university has, who sits on the panel, and if you're interested in certain professor, then you can ask them about the professor as well. So this was my result. So I tried CSIR like 3 times, and I was only able to clear the LS with the rank of 34. But with GATE, I was able to get my 199 All India rank. And with GATE, this opened up the opportunity for me to apply to in IITs, ISRs, even certain institutes, other institutes like BITS and all. So this was how the interview and written exam process works after you have cleared your fellowship exam. So most of the universities, if not all, they have a written examination even after you clear your fellowship. And in certain cases, they allow you to skip it if you have cleared with a good enough rank, or if you have a CSIR. So it depends on the university. If they want you, they can ask you straight to come to the interview. But for whatever universities that I went to for the PhD interview, they always had a written exam. So after a written exam, then they have two rounds or one round of interview depending on the university, and then you are shortlisted for your PhD. So the research interview usually happens, and it is kind of an intimidating tactic, I think. So what will they do is they'll make you sit in the center, and usually there'll be like a panel of 10, 12 professor. They'll be sitting right in front of you, and there will be only one light which would be on in that particular room, and that light would be shining right on you, and everybody else would be kind of perpetually in a darker space than you. So, when you enter the first time, it's a little jarring, but then the professors are nice. They'll make you relax. They'll ask you if you want a glass of water. They'll ask you a couple personal questions just to get you in the flow of talking. And then they'll ask you different kind of questions. Most of the questions that they're trying to ask is just to understand how much you know and what kind of confidence do you have when you're speaking to people who are experienced in that particular field. So their questions can land anywhere from what is genetics to what kind of pen is used in space. So there is like literally no say, or no pattern that they follow. They can even ask you to solve a particular question on board. So you have to be in a zone where you are aware that these kind of questions can come in, and you are prepared enough that if they ask you to do something like that, you can confidently go up to the board and solve that particular question. Then after your interview process, the first round of interview, they'll have a session with the professors, in which all the professors will tell you what kind of work they do. And then they'll ask you what is your choice of lab that you want to work on. Now this could be one lab, you will be like super interested in certain person and you want to work with them at any cost. Or this could be a list like 3 4 5 professors. So eventually what they do is they match your interest with that of the professor. And if it coincides and perfect, then you guys can both work together. But if it doesn't, then there is an option of moving to the second choice or the third choice or fourth choice, or how many ever choices they would ask you. After this, there would be a second round of interview. If there is only one round of interview, the introduction of professor happens before the final round. And then eventually, if you clear that, then they'll match you with the professor and you are able to move on to your PhD program. The PhD now consists of the first 6 months as it's basically a review of your master's. So they have, it's called coursework. So they have different subjects that you have to go through. And during the first 6 months, you are also introduced to what kind of work happens in the lab. So you'll be doing your coursework as well as your lab, and you'll be figuring out the kind of problem that you have to work on, both by talking to your professor as well as the students that are already present in the lab. So this is how your PhD journey can start. When I joined IIT Kanpur, which was in 2015, I was, of course, very excited, and we had bunch of plans with my professor. We were trying to work on a virus, which is adeno-associated virus, and we were trying to get into, trying to figure out how to decrease the immune response within the cell. And we were in the middle of solving the problem, and certain things beyond my control happened, and I had to leave IIT Kanpur. And that during that time, I was like, things are happening, I don't know how we are going to move forward. And at that time, it was, of course, very very sad. And I was also feeling very dejected, and I was like, how do I move forward? But then, I had a very supporting family, I would say, and everybody was like, it's okay if you are not able to finish this, you can move forward with something else. PhD is not the only thing. And at that time, it felt like, why is everybody telling me this? But then, when I'm sitting down here right now, looking back, I think, moving forward from that chapter was the best thing that I could have done. So, this would be my advice. If certain things happen in your life and you think that what you wanted is not working out in your favor and you have to leave it to move on to other things, it is absolutely okay. There is no reason to feel bad about it. There is no reason to doubt yourself. There is no reason to think that, oh, this is not working out in my favor. How are these things going to work? So, these kind of setbacks, they are really important because they teach you a lot about how life can progress. So if you have to learn how to persevere in certain conditions, and you have to move forward, having confidence of thinking that if it's fine, if this is not working on, maybe we can look at something else. That's why I have mentioned that, the PhD journey is not a sprint. It's actually a really long marathon, and it's full of up and downs. It's not a straight road. You will have a lot of things that you will be working on which will not work out. And then you will try to move on to other things which eventually will end up working out. So, so as I was mentioning, this was the work that I was trying to do at IIT Kanpur, and a junior of mine ended up following my work, and she did a really good job in figuring out how this AAV trafficking works using kinesin. So the hypothesis that we were working on was that there is a motor protein called kinesin which helps move stuff inside the cell from the cell wall. So when a virus enters the body, the immune reaction is generated. So there is a particular time frame that the cell takes to identify that it is a foreign molecule, and then it ends up generating an immune response. So what we were trying to do is we were trying to incorporate um, this kinesin molecule inside the AAV, so that instead of taking, say, 90 seconds to reach the nucleus, it took like half the time, like 45 seconds. So if you reduce the time, the immune generation also reduces. And this is important because AAV is used in gene therapy. So we were working on using AAV to deliver drug for rheumatoid arthritis as well as osteoporosis in patients. So we wanted to reduce the immune response because this is a helpful virus and not a bad virus. So of course, and the body has no way to identify helpful and bad, so it is going to generate response regardless of any virus. So we noticed that if we were to reduce the time for the AAV that it takes from the cell wall to the nucleus to deposit our gene of interest or the drug, it would reduce immune response. So this was what I was working on. We were trying to create a clone, and we were in the middle of cloning using siRNA knockdown approaches. TA cloning, um, and and then I had to move out of it. So when I moved out of IIT Kanpur, I was like, you know what, let's just take a pause, think through choices, and I ended up getting into a detour, which was my the pathology lab. So here in this is a pathology lab in Delhi, which is called Star Imaging and Pathology. In this, I was basically involved in marketing. So this was like a completely new domain for me. I had never in my life done marketing before. So what they wanted me to do was, they basically had me at as a speaker between the marketing team versus the pathology team. So there was new tests that they were designing, new instruments that they wanted to launch in the market, and they didn't have anybody who talked science and was able to also communicate with the marketing team at a level that they understood, because they had literally no background of sciences before. So they had me conduct tutorials. They had me conduct a kind of marketing materials that needed to be designed. They had me conduct CMEs, which was for the um, local doctors as well as hospitals, so that we can introduce our products with them. So there I understood that since we are in the science from from when we are like 18, certain things or certain language is already incorporated in our language. So when we are talking, we don't realize the person who is not from this background doesn't understand a single word of what we are saying. So when when I was speaking to the people who were working in the marketing team, I realized that they were also from a varied background. Like some was from art, some was from commerce. There were some people who were also from science background. But since they chose to do marketing, their vocabulary in science words was very limited. So for me to say that, for me to tell you, I was working with siRNA, they'll not know what siRNA is. We have to explain it to them in a language that a normal general person can understand. Even the people that we are trying to sell, we're selling the test or selling the equipments to, though they are doctors, they are expert in their field, so they also don't sometimes understand the science behind the particular product. So this was the first time where I realized that what I have learned versus what I have to communicate are two very different things. So if I learned something about a technique that, okay, say for example, uh, the product that they were launching is Biofire. What it does basically is it takes in your blood sample and it sees certain pathology or certain markers, you can say, and it checks the marker against a database, and it gives you, if particular marker is upregulated, then it can give you that this is the disease it has already seen it present in the database. Since it's upregulated in this blood sample, this particular disease is being, is in that patient. So now I understood the mechanism, I understood the science behind it. But when I was trying to talk to the people in the marketing team, they didn't care about the mechanism. All they cared about was how many samples in a day can we make this machine run? How many doctors can we approach in a particular day so that we can have the sample done? How much time would it take for a particular sample to run? And if can we run more than one sample at a time? Can we run two samples at a time? They didn't care about the science. They cared about the marketing of that particular product. So even if even if, and I realize that even if I know the science behind it, I also need to sell the science, right? And science doesn't just sell by just telling them certain thing is working in certain way. It has to have an impact on the society. It has to be important enough that a certain person who's also not from the science background needs to give their input in, and should feel that, oh, this is very important for me. So this was the time I realized that communicating science versus knowing science is a very different, two different things. And I learned how to communicate science during this time. So this particular time period in star imaging was really refreshing for me, because I learned that different ways I can use my knowledge and make sure that everybody else understands it too. After my star imaging, we are moving on to chapter 4, which was my PhD at Amity University. So there I worked with bacteria. So I used all the expertise that I had gained during my master's, my bachelor's, even the 2 years that I did at IIT Kanpur, and I worked with bacterial samples. So the research question was, why did we use bacteria and why the lipids? So we noticed that, and of course, this is now a more prevalent problem nowadays, that India is spending a lot of money on importing crude oil, basically from Russia and other the UAE countries. And of course, the fuel is a finite resource; it's not an infinite resource. It eventually will run out. So, how do we figure out a more sustainable or a renewable model? And we noticed that there are certain there are certain organisms which do end up storing lipids more than what they require. So basically, they end up storing lipids in a scarcity situations where they want to survive in a condition where there is, less food, less water, maybe the temperature is not right for them. So they end up
Okay.
storing lipid instead of using it. So these were called oleaginous microbes, and they people have already worked on a lot of them, including algae and fungi. But a lot of research didn't go into the bacteria, because because they're small organisms, they may not store as much as algae does. But then we notice that there are certain advantages in using bacteria as well, because their growth rate is superb. They can grow literally anywhere with any kind of component. And if they are able to grow in certain conditions which is not fruitful, we can use these bacteria in a real life situation where certain conditions are present in the environment, so that we can use that and create a good product out of it. So during this time, we used Indian soil samples from different states to collect the bacteria basically. Uh, so we had sorry, uh, I'm sorry. I can hear somebody from the background. Can you please mute yourself? Thank you. So we went around different states and we collected soil samples from areas which were not affected by industrial runoff or any kind of pollutant. We tried to find some places. It was a little difficult, but we did end up finding some places which are free of all the pollutants and all the chemical pollutions and human waste and everything. And after collecting the samples, we isolated bacteria from it, and during this time, we were able to find three different strains, which ended up storing lipids inside of them, which was more than 20%. And we used a response surface methodology method, which is a statistical analysis tool to optimize their growth conditions. And then we ended up using GCMS to identify what kind of fatty acids they have, so that if you were able to convert the fatty acid to biofuels, would it have good enough property? Would it have a comparable property that is already being used in the market, which is your crude oil? And this was the kind of questions that we were planning to answer during my PhD. So we found different soil. One of the main soils which I noticed was the cleanest, I would say is the soil from Kerala. And out of all the states that we collected the soil, we were able to isolate around 162 bacterial strain. To screen these strains, like whether they produce lipid or not, there is a methodology called screening with Nile red, which is a dye. It basically glows under fluorescence if a particular organism has lipid inside of it. So if the bacterial growth has fluorescence, if it has lipid, it will fluoresce under UV. And these are the strains that we take for further screening. So when you extract lipid from these particular strains, I found three strains which were producing lipids over 20%. And after optimizing their growth conditions using a research surface methodology, their lipid production also increased significantly. So now for identification of these three strains, we sent all of these samples for whole genome sequencing, which is basically 16S rRNA sequencing. And we found that all of the three samples were from Bacillus genus, but the species were different. So first one was Bacillus valenciensis, second one was Bacillus teculensis, and the third one was Bacillus pumilus. So all these three strains, thankfully, were not pathogenic. Otherwise, we would not be able to work on these strains. We would have to find something more. So all of these strains, they are well-known strains. They are mostly found in dairy products, during curd fermentation and all. And they have been used in other places. One of the bacteria, Bacillus teculensis, have been seen to have antimicrobial properties as well. So a lot of people have tried to use this in trying to curb infections in plants and trees. So these were good bacteria that we thankfully were able to figure out and find out. And then when we identified its fatty acid profile using GCMS, we were able to see that. Um, I'll just get back to it. Sorry. So we were able to see that all the fames that we had gotten was comparable to already the biodiesel standard which is present in the market today. So the percentage of saturated monofatty acids, polysaturated monofatty acids, unsaturated fatty acids, all of those were comparable, and those things were giving it a decent decent biodiesel characteristics, so that if we want to explore this more towards the industrial side, we can do that. So these three strains were the ones that I worked on. And we also noticed one thing, that the kind of food that we give the bacteria. Similarly, the way their fatty acid profile changes. So as you can see, the middle one, that is the A2, it grew very well in sucrose, where instead of glucose. So, when you are going through your PhD, you get to learn a lot of things. You'll figure out, oh, this is working better than something else. So, you have to open all of your avenues to figure out what is the best way forward. So now, when we did end up finding these three bacteria, now the question was, how do we make it sustainable? Because everything costs money, and if you're trying to get into the industry part of it, then you need to reduce your cost, so that when you're selling it to the customers, it is not at a very high end point that nobody is able to buy it. So we worked with industrial waste. So we saw that sugar industrial waste from a company, which like discards millions and gallons of waste every year. If we were able to utilize that waste, then we won't have to spend the money for giving it food such as glucose or salt or nitrogen sources. So when we worked with industrial waste, we saw that one of our strain, which was TA1, was able to utilize xylose in the sugar industry, in industrial waste, and was able to produce lipid. And the fame profile of that lipid was also really good, and it was comparable to the profile that is already being used in the market, which is the biodiesel standard. So this was a good approach for our work, because we were able to find the bacteria, we were able to use that bacteria on an industrial waste, and it was able to produce lipid which could translate into an industry work. So all of this work culminated at the end of 2025, where I was able to get my degree. And just a brief mention that during my PhD work, we ended up having COVID, and our lab was shut down for like a year and a half. And I, not just me, all of my like my colleagues as well, they lost so much of data. The 162 bacterial sample that I had mentioned previously, that number was even bigger. We had over 250 samples. A lot of strains died, lot of chemical got wasted, and basically it pushed us back approximately 6 months after we joined the lab again. So, as I said that, during your PhD, a lot of things can happen. And COVID was one thing that nobody has predicted, and we had no backups. We didn't know that, there was going to be a time when we are not going to be in lab for a whole year. Um, and, even to check up on the lab, because in the first 6 months, even that was not allowed. So after 6 months, when we went in, we saw that, the minus 80 was not working, all the samples were everywhere. And since we are working with bacteria, there was like contamination literally in the entire lab. We had to clean the entire lab, we had to fumigate it. Certain equipments just stopped working because nobody was using it for a year. So, all these things happen during your PhD. And of course, I don't think COVID kind of situation can happen again, but then, you never know what is going to happen. So you have to be prepared for everything that can go wrong, and also have backups in place, so that if in case something does go wrong, you are, in a safe place. Um, but as I said, some things are beyond your control. So I would say that control the things that you can control, and just leave everything else. um, because if you are worried, or if you're anxious about things that is not in your control, then you will not be able to enjoy the good part of it. So even though, it took me like close to 6 years to finish my PhD, but then the journey taught me a lot. It taught me, that you have to be strong, you have to be optimistic in certain situations, and sometimes you just have to leave certain things to the higher power, and say, okay, fine, if it is not working, we'll just see how things will progress further. So this was my PhD. And now coming to after PhD, how things look like. So I say that PhD gives us the superpower. So this image is by a very influential person to me. Her name is Elena Hoffer, and she runs a company called Alma. And she had put this picture up on her LinkedIn, and you can see that as a PhD, people think that we are just a student, right? Okay, we are just doing a research, but we are not. We are literally, a concoction of all of these things. We have designed images, we have ran projects, we know how to submit bills for certain certain transactions. We have taught, we have edited papers, we have reviewed articles, we have analyzed, we have done statistical analysis, we have done bioinformatics work. We have tried to figure out how to, if something breaks down, how to fix that without actually involving anybody else. We have also spoken at different platforms. We have went to conferences. We have spoken on series like, like Biopractify is doing. So it's just not research that we do during your PhD. You are literally learning so many things. And when we are trying to market ourselves, we tend to forget all of these things. That, oh, I just did my research. No, we didn't. We actually went through a lot of phases that people take so many years to learn, and we ended up learning everything during our PhD. So that is why I call it that PhD is your superpower. You are just not a researcher. You are a lot of things, and you can use those particular techniques to market yourself if you do not want to get into the academics. So there are a different avenues that you can explore after your PhD. The first is the normal one that lot of people go through, that you do your PhD, you do your postdoc, and then you ended up teaching or becoming an assistant professor in a university. There are also places that you can work, like pharma and biotech companies, which have a dedicated R&D department. They have where you can use your expertise in your PhD and directly apply your work there. So that is one other approach that you can use. Nowadays, the third approach is scientific communication. This is coming up a lot, because you want the people to know what research is going on, how things are progressing. So you want to communicate everything that is there and publish it to the world. So science communication is also a great place that you can explore your expertise in. Then we can come to areas which are regulatory affairs and um, patent analysis. So both of these things, even though you may not have studied patent analysis in your PhD or not done it, or may not have a patent, you can appear for an exam and become a patent analyst, and then you can apply to certain places in government offices as well, and you can apply your knowledge in your science and approve or disapprove patents accordingly. And the fifth avenue you can go that I am trying to get into is entrepreneurship, which is, of course, start your own business, build your own product, use the knowledge that you have gained during your PhD year as well as your previous years, and trying to create something that people can use. And the sixth, of course, is your government and policy offices. We can go for advisory role. You can go for policy places. You can also apply for public health agencies. And then, these are kind of things that you can get to explore. After your PhD. So your journey doesn't have to be pure academics. It can go into a different direction. And whatever suits you best, you can go ahead and do that. Now moving on to my work that I did, which is Simplify Research. So as this was the problem that I understood when I was doing my like star imaging and path lab work, that communicating science is very different. So I noticed that when you are starting your journey into sciences, the papers that you have to read in order to understand what you want to work on is so knowledge dense or information dense that sometimes things just don't make sense to you. So I wanted to design something that would help minimize that headache and give you a better knowledge or better idea of what one paper is saying. So I ended up designing Simplify Research. So basically, this is an AI tool which reads the academic research paper for you and breaks it down into different explanation, which is a summary, explain it to me like I'm 15, and a technical deep dive, which would have all the main core science works that is there in the paper. What would be the industry relevance, a methodology, and a report which is downloadable. So my main focus was students who were starting in the sciences, as well as who are, of course, well versed into the science also, because sometimes paper are just too long, and then, um, you want to work on it and you are not able to understand it. So how I built this work. So basically, I started. I'm sure you would have known Emergent. So Emergent is an AI which develops websites for you. So you give it certain prompts and it will create a website for you. What it does not do is that it is not able to incorporate a lot of data, and like as a person who is designing a website, I have no control over the data that is being inputted into the platform. So I wanted to do something which would give me full on control over my work. So I took the code from the emergent after designing a basic layout, and I used Claude to create a front end and a back end separate from the emergent, and that would get straight into the website that I have created. So of course, Claude, let me just clarify, I have no knowledge of computational at all. The basic computational I know is few bioinformatics tool, and what statistical analysis I had done, a little bit of R, and that's about it. So pure coding and all of those things was not ever in my forte. So, but considering the AI has now boomed like crazy, and things that you were just planning in your head can now gotten you can write down on a piece of paper and have it done by an AI platform. So that's what I did. So I used Claude and I created this particular platform, and I told it that these are kind of things that I want it to work as, and it gave me different codes to run it on. And I learned how to create a repository on GitHub and connect that repository to the backend and the front end servers, how to create a database so that when somebody is submitting their information in my app, that that data is not leaked or it is private. And so all of these things I learned while I was developing my tool, which was Simplify Research. So, you can see the tool today in front of you, but the back end process of it is really, really long. You have to take care of a lot of things. You have to also ensure that, if certain people are using it, so it's not crashing, or you have enough space for the all the information, the data is saved properly, it is not going anywhere, the only person who can access sorry, the person who is able to access doesn't feel like their data is being breached. So all these things get into it when you're trying to develop a software. Now when you have developed the software, it I can if I can just show you guys the in the can you see the screen?
Yes, ma'am.
Hold on. Sorry. This is how the end product looks like. Okay, so here you can upload a research paper and it will analyze your paper for you. So I have just prepared one paper for you guys to see. Um, so this is the paper. It is about gastric tumors, and it analyzes the paper. Yeah, yeah. Oh, yes, there. It has analyzed the paper. I think something is going wrong with this. Sorry, 1 second. Okay. Okay. So this is the paper for one of the condition that I was working on, just stiff person syndrome, and that's how it develops a summary. So it will give you a paper summary which would be like around 150 words, and it will tell you what the paper is talking about. It will also give you a basic explanation that this paper describes a rare brain disease where immune system attacks the proteins. It will give you the key takeaways, and it will make you understand the paper in a very basic ways, that what happens, in the brain, if it is comparing it to like car with the broken brakes where muscle can't relax properly. So if the paper is information dense, and if you are trying to figure out how things work, then this is a way. Then I designed the software so that you can understand the paper. The next part is the technical deep dive. It will give you objectives, what kind of methodology the researchers have used, the result that they have, and the conclusion that they have. So then it can focus on the industry relevance, like why is this work important? What kind of things that they can do? What real world application is going on currently on that particular disease or the particular work that the researchers were doing? The roadmap that they followed, like what is the future prospect that they designed, the limitation of the work, or what can be the future things that you can do. So this particular part I designed because when you're trying to work on a research topic, right, you need to figure out the gaps, and it can tell you the kind of gaps that a particular study has. So you can focus on these gaps and figure out if somebody has worked on it already. And if not, then this is something that you can explore if you want to do this work. And then finally, it has a methodology flow, which breaks down in each step what the researchers did and the kind of finally the results that they got. So this was something that I worked on. You guys can explore it as and when you like. Um, I just will go back to my PPT. So this is the work that I did. The last thing that I would like to mention is that, during your work in your PhD, one group of people are really, really, really important. That is even more important than the kind of guide you have or the kind of university you chose to, and that is your peer group. So this is the work. This is the people that I work with. These are all my friends from my bachelor's to my master's, even my IIT Kanpur people, Amity, and the people that I'm working currently with. That is Next in Biology newsletter. I think you guys would have heard about them. They are really amazing science communicator people who are just doing explaining science to everybody because they have the love for science, and working with them has been so refreshing because after working in the lab for past 6 years, when you're trying to sit down and work with different people from different background, you get to learn so much more. Like, I mean, I have enhanced my technique, especially in illustrations, after working with this team. So they are amazing, amazing group of people, and all of them are doing this voluntarily. There is no monetary involvement, there is no nothing else, just the love of science that they are trying to portray themselves, that they're trying to portray to everyone else. And they have articles published, and everybody is working super hard, and amazing people. So your peer group is very, very important because at in a lot of stages, even though your family is in your support, but they don't and understand what is going on currently in your lab. So these people are working with you, they are your friends. You can vent out to them, your anxiety levels can go down after you just go for a nice tea session with them. So everything else is of course important, but the kind of people that you hang out with is very, very important. They are your lifelong people. You can, I am still in connect with almost everybody. So they are your lifelong friends. So you have to make sure that your peer group is really, really strong, so that when things don't go your way, you have somebody to just go and vent out to. So this is what I wanted to take away for you guys, that the PhD path is not linear. There are things that are going to go in your life that are not in your plan, and it might feel weird, but then things end up working out. All the tests that you are planning to, they don't judge you for your worth as a scientist. They are just so that there are so many people trying to get into a PhD. It's basically a barrier that you have to claw across. So don't undermine yourself if you're not able to clear these exams. There are a lot of universities which even don't have these exams. You can go straight for their for those universities, and trust me, they are having good research work there as well. You don't need that tag of certain university. As long as your work is good, you're going to go places. Okay, the PhD is your journey, and it is just the beginning. Okay, this is the phase where you're just starting, your entire life is ahead of you. So don't think that PhD is the end. No, it's just the starting. Once you're done with your PhD, your life is going to begin in a new way. Okay, with the AI tools, you don't really need any knowledge of tech as such to build a product. So if you are, if you have a passion for something that you want to work on, I would suggest please use these tools that are available in the market nowadays, which were not available when we were doing our PhD, and just learn and try to build a product that you can, market properly. Of course, and the most important bit of after research that I have got to know is that communicating research is very important. You have to be able to, tell what you have worked on without overwhelming the person who's listening to you. Okay, so thank you, and if you have any questions, feel free to reach out, please.
Um, Hello, ma'am. Am I audible
Hi.
to you?
Yes.
Ma'am, so I'm a third year BSc student right now, and
Mhm,
I do find my interest. I do find continuing till my PhD. I want to continue in PhD, but
mhm.
and also I want to continue research after
Right.
PhD.
Mhm.
But today I was meeting with few people. I had a webinar today, one more webinar, where someone told me that research field is not financially lucrative. It is like we have to struggle financially always. There are no jobs and like this.
Those see, I understand your apprehensions. It is, and somewhere sometimes it does tend to be true. Financially, if you go straight into the academia bit of it, like if you end up doing postdoc and then you go into assistant professor, it is a long journey, right? And a lot of postdocs are dependent on government funding. So if you're going into a university which has a lot of funding, you'll be fine. There will be no problem. But you have to get there to end up getting that funding. So if you don't want to do the traditional work, I would suggest look for industries. Industries have a lot of openings. They have R&D department, which will, continue your love for research. You'll be able to handle projects, and money is not a problem there. Okay, so you are in India right now, right?
Yes, ma'am.
Yes. So look for different industries. In like what kind of work are you doing? Sorry.
Ma'am, just a third year BSc student right now, BSc Life Sciences.
Acha, okay, your license. So look for industries which are relevant to your work. Okay, don't go for anything and everything. Anything that is in relation to your work, be it pharma, be it some normal R&D place, they will have an opportunity for you. And see, when you're starting, a little financially trouble will be there, but once you're into the industry, then these things don't matter. Like I'm in the US right now, and I have people who have worked in particular companies for like 5 6 years, and their salary is amazing. Okay, so initial 3 to 4 years there will be a challenge once you're trying to establish yourself, you reach a certain position, and then it should not be an issue. So don't be disheartened if somebody says, that PhD is not going to give you money. It will just take time.
Thank you, ma'am.
Yeah, no problem. Anybody else?
Hi, ma'am.
Hi.
Hi.
Hello.
Hello. Yes. Hi.
Ma'am, I'm a MSc first year student.
Mm-hmm.
Now, yeah, I'm planning to apply for dissertations out here in some of the central universities. But the thing
Mm-hmm.
is, I am studying in a private college. So
Mm-hmm.
the dissertation window that they are giving is about 4 months. So like from Feb to somewhere around July. So
Mm-hmm.
will that be you know, a valid period to give obviously I have to give it now. And how do
Yeah.
I approach institutes and how will that work, ma'am?
Yeah. So, if you're not able to do your dissertation in your university and you're trying to apply somewhere else, this problem does occur. Sometimes certain universities say you are getting 4 months. I knew a student who was working with me. She only had three. So it's a common problem, and the professors also know about it. So when you're trying to apply, wherever you're applying, just drop a small email to the professor initially that, I am working on this and this work, and I'm from this and this university. And one constraint of my university is that, I have to be back in my university after 4 months. So, do you have a project that I can work on for 4 months? This is a list of expertise that that I have, and I can help you in this particular work. I would also suggest you to do a little bit of research on what kind of work that particular professor is doing before dropping them an email. Okay, so 4 months is okay, it's fine. I mean, they'll give you a project that you will be able to finish in 4 months. It should not be a problem. 6 months is just 2 more months. I mean, it's fine if you don't have those 8 weeks. Um, just drop them an email. Tell them, even like LinkedIn is a very good tool right now. You can even approach students who are working in the lab. But you have to do a little bit of research. Not all institutes will agree to the 4 month policy because they would want you there to be there for 6 months. But there are a lot of institutes that do have that, they have no issues. BHU being one of them, because I know there was a student there for 3 months. So look through the professors who are working there, drop them an email, and more often than not, they end up replying to you. So that should not be a problem.
Thank you, ma'am.
Hi, ma'am.
Hello.
So myself Latika and I'm from Kerala. Ma'am, I'm doing my third year in BSc Biotechnology.
Mm-hmm.
So here it's been a 4 year program right now, BSc and BA and all. So my confusion is still that should I continue fourth year? Uh, After finishing 4 year, we can do MSc for 1 year. But I want to do 2 years proper MSc. I have interest in genetics related subjects like cytogenetics or epigenetics. So, what should I? I'm still in a confusion, ma'am. So, could you clear me that?
So what is the fourth year that you will be doing after this year, after your BSc? Like, if you're not, what is the fourth year that you'll be working on? Like, what degree will you be getting?
So, ma'am, after fourth year, we'll be getting a BSc degree with honors.
Oh, okay, 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 okay. So, if you are really want to get into the masters, then it doesn't matter if you have a honors in your bachelor's, right? The more important thing is how are you able to clear the exams and get into the counseling session with the universities. So, if you are really, really sure that yes, you want to go ahead and do the 2 years masters, then there is no need for you to continue for one more year, because in any way, um, you know, um, you are going to spend 2 years for your master's degree, and that would be more subject specific to you as well. So honors is fine. I mean, it is, if you don't have honors in front of your bachelor's, it doesn't really matter eventually. So you can go ahead and focus on the universities that have your cytogenetics and epigenetics, and apply for those universities for your masters. And it would also be a good thing to apply directly, because whatever you have studied in your bachelor's would be fresh in your mind. So when you are trying to get through these exams, it would be a bit easier for you in if you were to take a break instead. So it's fine. You can go ahead for your master's, not an issue.
Okay, ma'am. So, will that, ma'am, I have a wish to study abroad. So, will that affect this too?
With studying abroad for masters, um, so see, usually they have these what do you call it, equivalency places. One of them is WES. So what that might affect your work a little bit, because if you have a degree with honors, they equate that to their 4 years programs of bachelor's. Okay, I am not sure if like 3 year BSc program, what will they equate it to? So what you can do is, when do you have to give this decision?
No time limit, ma'am. They haven't reported us, but they are asking
Okay.
for.
So do one thing, find this equivalency site. WES is one of them. And if this is for US and Canada specifically, I'm not so sure about the Europe bit of it. So drop in your transcripts there and see what kind of equivalency programs that they're equating it to. So if your 3 years program is not equal to their bachelor's, then you will not be eligible for their master's program as well. So if you want to go abroad, then the safe bet is to go for the fourth year. But if you're trying to do this in India, then the fourth year is not a problem. Then you can just go ahead after your
O-
3 years for your master's.
Okay, ma'am. Thank you
Okay,
so much, ma'am.
no problem.
Hello, ma'am. Am I audible?
Yes. Hi.
Hello, ma'am. It was a great session. Thank you so much for this. It was really
You.
inspiring. I have a small doubt to ask in regards to your PhD. This is a
Mm-hmm.
very small doubt, if you may.
Yeah.
Uh, The microbes, to produce more lipids, I wanted to ask why you did you not look at the nutrition in the first place? Or maybe if you
So
did look at the nutrition in the first place.
yeah, so nutrition, I would say what I focused on was I wanted to minimize the nutritional cost at because putting in money for glucose, your nitrogen sources, even your salts for that matter, is going to, eventually give you, it's going to be in a lot of money. So I wanted to make them grow in a scarcity because of course when they're growing in scarcity is when they're going to store the lipids. If they're growing in a normal environmental condition, they're going to use that lipid to reproduce. So when you are trying to grow microbes in scarcity, they only need three major nutrients for that matter: carbon source, a nitrogen source, and salt. All the other nutrients, I made sure that they didn't have any of it. So when you give them basic nutrients, what they do is they utilize this nutrient to produce the lipid and store it so that when they get into a normal environment, they are able to utilize lipid for energy and reproduce and then, do all sorts of things that they do. So nutrient was never a focus. The focus was to deplete them of nutrients so that they'll be able to store the lipid instead of using them. So that is one I can say a balance that you have to create that you don't deplete them so much that they die and you don't give them too much that they don't store the lipid. So you have to figure out that balance.
Okay, ma'am, I understand now. Thank you so much. It's because
Problem.
I'm working in this similar
Mm-hmm.
like microbe industries that I'm getting to know more of this. And also, ma'am,
Oh,
if you
okay.
may, could you
Mm-hmm.
please share the link of the website, Simplify Research?
Yes, I can drop it in the chat for everybody to explore. Give me 1 second. Okay, you guys can wait. I think I misspelled something. Okay, let me do that again. Okay, go for the second one.
Okay, ma'am. Thank you so much for
No
this.
problem. Thank you.
Hello.
Hi.
First of all, thank you so much for this excellent talk, Dr. Sugandha. Very inspiring journey, I must say, and it has been an absolute pleasure to work with you alongside in Next Gen Biology International newsletter. And as always, you inspire a lot. So I just have a bit of personal question to you. So I mean, I did type it out in the chat box as well. So, I mean, can you just share a quick research failure that ultimately became one of your greatest learning experiences?
So the biggest the biggest one I had was when when after coming back to COVID and realizing that most of my samples were dead and there was no way to revive the bacteria no matter how strong they are, but eventually if they don't get food they die off. So coming back from that was a bit of a bender, because also during that time I had a baby and he was like 6 months 7 months, so I had to manage the lab along with the kid and everything was a bit of a mess for a few months. But then um, you know uh, I what I do is when I'm stuck in certain situations, I tend to write things down. So what I did was I made a note to myself. I was like, okay, these things are working, these things are not working. So if these things are working, let's move in the direction of the things that are working. And for now, ignore everything else, because when they are not working and you keep looking at it, your brain tends to focus more on the negative. So I just, used to ignore those things and I have to work on the positive bits of it. And of course, as I said, peer group is very important. So what I did was I called up all of my friends from different states and I said, I need a soil sample. Please figure out how to send it to me. And most of them, they literally posted me a big box of sand so that, I can isolate the bacteria again. So success doesn't always have to be pretty. It is going to be really, really messy. You are going to work yourself to the ground. You're going to figure out new ways to figure out issues. Because COVID was such a big shock to all of us, and we had, like, even the professors didn't know how to work around it. So everybody was just working together to figure out the instrument situation, to figure out the loss of data, the loss of bacteria cultures. And I was not the only one. There were like multiple people who were facing similar problems. So, working together is a good thing. Have a good team, have a good peer group. You'll work it out somehow.
Great. That must be a very terrifying experience, I must say, like seeing your samples being dead. And thank God, like you had a great team to work with.
Yes, I
And
was
also,
working with
I'm a bit,
bacteria. I mean, people are
yeah,
working with human samples. I mean, they were more devastated than me.
yeah, I see. Um, Also, I'm a bit curious to ask a question. I often ask this question to a lot of people. So, what true success means to you?
See, success at the end of the day is something that makes you feel good about yourself. It doesn't have to equate to money or a status or, an award or a degree. If you worked in your lab and you were able to, just figure out one experiment, like, one problem that you were able to solve, that is success. If you feel good about it, you're like, okay, I worked for 6 hours today, fine. If you feel good about I worked for 1 hour today, fine. Anything that makes you feel more confident, more good about yourself is success. It doesn't have to be awards, money, or reaching certain positions, something small is also very important. So that's what I say. It's that, don't worry about the awards, don't worry about the degree, just work. If you're loving your work and you're working, it's fine. I mean, that is what success is to me.
Great. Uh, Thank you. Thank you once again, Dr. Sugandha, for such an inspiring talk. Thank you.
Thank you, Charan. Thank you. Okay, I saw there is one question. I just opened the chat box by Varda. So see, the use of AI, I would say is a precarious ground for now at least. So what is happening nowadays is that people are using too much AI. Even if, see, you have to use it in such a way that it simplifies or eases your life, but not so much that you are so dependent on it that even to, write a paragraph or an email to a professor or a guide, you need to open Claude or Chat GPT. So use AI to make things, to make your life easy, but not so much that you are so dependent on it that, basic stuff is also gone out of your head. And I have noticed that, even with people who are working, like my juniors, that they are so dependent on AI that simple illustrations are also not able to, they are not able to create. What we used to do was, we used to sit with different softwares like PPT, BioRender. For me, Canva was a new thing. I have never used Canva before, so I was learning how to use it too. So don't like let AI run your life. You run the AI. If you're using in, trying to understand something, yet, okay, fine, I'm not understanding. I have nobody to speak to. There is nobody to help me out. Sure, go ahead. But if what you understood, if you are to copy paste it and write a research paper out of it, that would be wrong. For your understanding, it's good, but for you to copy paste it without ingesting the knowledge, I would say it is wrong, because then you are not learning anything. You are just copy pasting. And research is all about learning, because if you don't understand how basic methodology of something would work, you will not be able to present it anywhere else. So if I were to ask you the principle of centrifugation, and you are not able to tell me that what kind of principle it uses to, separate the layers, then how are you going to utilize that particular piece of knowledge for your PhD? Or for that matter, if you're trying to present somewhere in the conference. So AI can help you, but then don't let it run your life. Use it normally. If something is stuck somewhere, fine. But then, don't try to use it too much that basic concepts are also not, you're not able to clarify. So yes, for article finding, sure. Writing article, no. That's a big no. Article finding, you can shortlist your places where you can look for, but writing article, no. And I'm clarifying this now. A lot of AI, they will give you wrong links, so they are not up to date to the current research. So the best bet you have is go to PubMed, go to Google Scholar, and then manually do the research. That would be the best chance you'll have to find the best articles. Okay, never ever use AI for writing articles. Most of the journals that you're going to publish, they're going to give you a big no. They have different softwares in check for this. Even like minimal acceptance maybe would be 10%, because that sometimes is false positive. So they'll give you that benefit, but beyond that, they'll just outright reject your articles. Okay. All right. Anything else?
Good evening, ma'am.
Hi. Yes.
Ma'am, my name is Shashwati. I'm currently pursuing BSc Biotechnology. Ma'am, I have, it was, thanks a lot for the session. It was really good. And I have used Simplify Research, a website, and it was really so helpful for me. Ma'am,
Thank
this
you.
point, I'm not sure if I will be pursuing research in the future, but if I were to, I would like to ask you, ma'am, could you share some common misconceptions that you think undergraduate students have regarding research, which we can correct it?
When you're trying to figure out if you want to be in the research, the common misconception is that, you do your MSc and then you do your PhD and then you have to do your postdoc. So everybody tends to forget that there are other avenues to explore other than the common one. I have known people who have done masters and they have gotten straight into an industrial job, and they have never pursued PhD and they are quite happy with their work. So one of my friend, he did his masters and then went straight to TCS, and then he's been working there ever since, and it's been close to 7 years now. And I asked him that, why did you not try to do PhD? And he was like, I have no interest in research. What am I doing? What am I working on? I'm more than happy to it. There's one other person who she did her master's, and she joined Elsevier as the scientific writer. And she has been working there for close to 5 years now. And she never thought that, PhD is an option. So if you don't want to explore research, it is not the end of the world. There are so many things out there that you can explore. You can go straight for that. I mentioned patent analysis as well. You need, I think the eligibility is just a bachelor's or maybe a master's, but just to check once. And then you can go for patent analysis. You will be in the field of science, but you'll not be doing research. So, don't be in a state that, just master's, PhD, postdoc, that's the only way. There are different things to do. There are a lot many things. And trust me, from the time that I have started my PhD till today, there are 10 more things that have happened that even I may not be aware of. So it is developing at such a fast rate nowadays, AI being one of them. Um, you can explore that as well, science and AI, how these things work.
Okay, thank you, ma'am. I'd also like to take this opportunity to thank Biopractify for organizing this. And also, I got to know about Simplify Research from Next Gen newsletter. So, thanks to them. Thank you.
Thank you.
Most welcome. Uh, Thank you for your inspiring and insightful sessions. Your journey, valuable guidance, and practical advice have motivated us to pursue research and confidence. We truly appreciate you taking the time to share your experience. I wish you continued success in all your future endeavors. Thank you once again.
Thank you so much. Thank you for having me. And thank you for being a great participants, guys. Thank you for listening to me.
Okay, so we will just wrap up our session for today. Thank you so much once
Thank
again and have a
you.
great day.
Same to you. Bye bye.
Thank you so much. Thank you.
Thank you, ma'am.
Thank you.